First, we have Dr. Khanh Kieu. Khanh Kieu is an associate professor at the College of Optical Sciences, the University of Arizona. He received a Bachelor of Science degree in Applied Physics in 2002, followed by a Master's in Laser Technologies in 2004 from St. Petersburg State University of Information Technologies, Mechanics and Optics, in short ITMO, Russia. Thereafter, he received a PhD in Optical Sciences in 2007 from the College of Optical Sciences, University of Arizona. During his PhD studies, he worked to develop new fiber-based devices. In 2007, he joined Professor Khanh Kieu's group at the Cornell University to do research in ultra-fast fiber lasers. In 2009, he came back to the College of Optical Sciences, Arizona as a research faculty. Dr. Kieu started his independent research group in September 2012. He is also the founder of K Photonics, a startup company supplying compact, low-cost, ultra-fast fiber lasers for research and education. He has authored and co-authored more than 150 peer-reviewed journal articles, 5 issued US patents, and has given numerous conference talks and proceedings. His current research interests include ultra-fast fiber lasers, nonlinear fiber optics, nonlinear spectroscopy, frequency combs, micromachining, microresonators, and biological imaging based on supercontinuum and ultra-fast fiber light sources. Please join me in welcoming Dr. Khanh Kieu.
So our college is part of the university, and is these 3 buildings right at the corner of the university here. I'm not sure if you can see my mouse, but if you look at the college, we have 3 buildings next to one another, and we're building a 4th building on the back side. So we're going to have a lot more space. So our college is located in the so-called Optics Valley, and we have about over 300 companies in optics here. We have many different areas of research in optics, including optical physics, image science, applied optics, and photonics, optical engineering and design. So on our faculty, we have Nobel laureates, we have a lot of people from professional societies and entrepreneurs. And we do everything from education, research, and outreach in all aspects of the science of light. So anything that related to light, we are interested in. So our college offers different degrees here, from bachelor to master to PhD. And we have over 30 undergraduate classes and 70 graduate courses, and then we have even online classes as well here. So here are the 4 main areas of research that we have in the college: optical engineering, imaging, which is very relevant to today's topics here, and then we have optical physics that cover from the fundamental physics, quantum phenomenon, to ultra-fast lasers, to computational optical physics. And then we have photonics as well. So we have things that related to telecommunication, high power laser, multiphoton microscopy, and so on. So these are the 3 main areas that we have. So these are the brief introduction to our university and our college. I would move on next to introduce my research group, which is the ultra-fast fiber lasers and nonlinear optics group. I would like to introduce my student first, because a lot of the things that I'm showing next, my student did a lot of it. Okay, so I would like to thank all of them for their contribution here. Okay. So next, I would like to briefly show you our research directions. We have 2 main areas here. The first one is laser development. So we built a lot of different type laser, working at 1 micron, 1.55 micron, or 2 micron, and even beyond. And these lasers are of different type. So the focus of today's presentation is on ultra-fast laser development. So I mentioned very briefly about these lasers that we built in our group. And then on the application side, we also have a lot of different topics here, but my focus would be on multiphoton microscopy, which is actually quite interesting, and we've been working on this area for many years. And I hope to share with you our latest results in this area. So first, I would like to just briefly mention about optical microscopy. In particular, fluorescence based microscopy, which is a state-of-the-art instrument for biomedical imaging. Okay? So you have some microscopes that look like this, and then you have on the side the excitation laser, for example, and then you can capture the image using the camera on the microscope. And this microscope, the requirement typically is that you need to stain your sample, right? So you need to introduce different fluorophore that would fluoresce when you excite them, okay? So traditional microscope needs stain to create contrast in biological tissue. So these are a couple of standard images that see with optical microscopy, standard optical microscopy using H&E staining. So these are the standard techniques that if you do a biopsy nowadays in a hospital, that's what your doctor or your pathologist will have to do to be able to visualize the cells and the structure in order to do a diagnosis. So I would like to introduce very quickly the physics of fluorescence, right? So you have a molecule that you can excite with a high energy photon, typically in the blue or in the UV. So the electron inside the molecule or inside the atom is going into the excited state. And then when it goes down back to the ground state, you have a red-shifted photon that is emitted. And you can detect this emitted photon in order to create an image, right? So that's a standard way. So now the next question that we can ask is that, can we use more than one photon, right? So in this diagram here, I show a scenario when we have two photons that being absorbed at once by the molecule or the atom. And then you also have one photon being emitted, but this photon is actually have higher energy compared to the energy of the photon that you put in, right? So this is a very unique piece of physics here, and it requires a very special condition for this to happen. So it turns out that this process is possible. It turns out that molecules or atoms can interact with multiple photons at once. And the requirement is that you have to have enough density of photon, right? So you can introduce this, you can enable this process. This idea was actually predicted a long time ago by Maria Goeppert-Mayer. When she was doing her PhD degree, she already wrote a paper predicting that this process, the process of two photon or multi-photon absorption should be possible. And only in 1961, Pfizer and co-worker demonstrated that they could have two photon excitation of calcium fluoride, right? Look at europium here. So you can see the laser that they use to excite the sample, and they can see the fluorescent emission as a result of absorption of two photons at once here. So it turned out this is the basis for modern multiphoton imaging that I'm going to talk about later. So it turned out that we can do a lot more. So the first idea was two photon absorption induced fluorescence. It turned out that you can do second harmonic generation, right? So you come in with two photon and you come out with one photon with an energy of two photon. And you can generalize that into three photons fluorescence or third harmonic generation. So these are the processes that we are going to use to create contrast for imaging that would not require any staining at all. As you will see that these processes will be very useful for medical imaging and diagnosis. So another process that for this audience I think I have to mention is based on basically Raman scattering. So this is a process that we have been working intensively in our group, but I won't have a lot of time to show you our result in this area. So basically you come in with two laser beams, right? So one would be the pump and the other one would be a stock. And once you have the right condition of the molecule, you can generate the so-called anti-stock signal. And you can use this anti-stock signal to image a sample. And using this Raman scattering effects, you can do chemical mapping, right? So now you use a chemical as the imaging contrast. So we can map our lipids, for example. We can map out a lot of proteins in the sample using this technique. So this is really powerful, and for today's presentation, I won't have time to go into detail because I would cover more on the multiphoton side, okay? So now, multiphoton microscopy was demonstrated, so the mature form of it in the early 90s, right, 1990s, by the group by Wattweb group at Cornell. So this picture shows the principle of the concept, right? So this is a cuvette that you have some fluorescent dye inside. So you take a continuous wave laser working at 488 in the blue, you focus it down to the cuvette, then you see the fluorescence, right? That's the yellow signal there. Everywhere where you have the laser beam, right? You can see the full cone of the fluorescent signal. So now if you take a femtosecond laser working at 960 nanometers, so this photon energy is much less than the band gap of the fluorophore, right? So you can see that only as the focus spot of the laser beam, you can generate the signal, right? Because at the focus spot, the intensity of the laser beam from the femtosecond laser is so high that you can have two photon absorption and then you create fluorescence that way, right? So now you have a very localized 3D type Voxel excitation. And now if you take this focus spot and you scan that in 3D, you can create a 3D image, right? So that's why multiphoton microscopy provide 3D sectioning capability. It is very non-invasive. So we have image sample that is even alive and you don't really burn or kill the animals at all. It's provide very high resolution because it's an optical technique, right? So you can achieve sub-micron resolution. And staining a lot of time is not needed at all. So we can get the sample from the biopsy, let's say from a patient, and you can that image right away. And if you use coherent Raman imaging, then this technique can have chemical sensitivity. But in order to do this type of imaging technique, we need a femtosecond laser for this. And femtosecond lasers are seen very expensive and quite cumbersome. So this is a standard setup that you can buy nowadays from different companies. So this setup, for example, would cost you over $500,000, right? So you have the microscope, you have the laser, and then you have to put everything together with the software, right? So it costs $500,000, and it's require very special training, right, to be able to for us to use. So that is why the main bottleneck for this technology to be widespread is the cost and the size and the complexity of the setup, right? So that is a problem that we have to address in order to make this more widespread. So first about femtosecond laser, this is the traditional state-of-the-art ultra-fast laser that you can buy from Coherent, for example, here. But they are really expensive. So a laser like this would cost about $150,000, right? And then if you want to extend the wavelength of your laser, you have to add in an OPO. That costs another $150, right? About $1,000, actually $150K, right? So, and it's quite bulky and it's difficult maintenance. So you need a person with a PhD degree to run this laser. So these are why these are very difficult to move out of research labs. So that's why we think that using fiber laser platform would be much better, because we have very high efficiency, we have very compact, and we don't need to do any alignment at all. But the challenge is would be very difficult to make this fiber laser that I show here, that I'm holding in my palm. Very high power is difficult, and mode locking to generate femtosecond pulses is very difficult, right? So over the years, we have created some solution here, and it seemed to work very well. So this is an example of a laser that we built in our lab, and this laser has been commercialized as well. So this laser you can hold in your hand basically, but it can deliver less than 100 femtosecond pulse duration, and then more than 100 kilowatt peak power. And then the cost is about 1/10 of the cost of the TISAP laser that I showed you earlier. So now the next thing is the microscope itself. So we actually design and build the microscope ourselves. If you look at the diagram here, you have the arrangement of the microscope, right? So you have the lasers that you use optical fiber to deliver the laser beam to a scanner here. So it's at the ground bore scanner. And then you have a telescope to expand the laser beam to fit the back aperture of the microscope objective, right? So, and then your laser beam is focused down on the sample. And then the signal that's being generated, right, based on multiphoton absorption, based on second harmonic or third harmonic generation, you would be detecting using these PMTs here, right, photon multiplier tubes. So this is how it looks like in the lab. That is a microscope that we built here. So you have the femtosecond laser, and you have the microscope here, and then the sample would be put under the microscope objective right here. Okay, so you know, once you have a microscope, it's actually a really fun thing to play with, right? So the first thing we play with is to use this microscope to characterize 2D materials, because these materials are really, really interesting materials that can have a lot of important applications. So these are atomic thick layer of graphene, let's say, of in this case, the sample of MS02. So we have 1 layer, 2 layer, 3 layer, and so on. And in the third harmonic signal, you can see clearly the different layers. And even on the second harmonic signal, you can see even a single layer of atoms, right? That on the sample here. And then students actually had a lot of fun doing this gemstone, right? So, or minerals of different sort. It turned out that nobody else have looked at these stones using multiphoton microscopy before. So we were the first group in the world to look at this. And if you can see here, we see a lot of interesting feature inside. And here's some of the other images as well that nobody else have seen before, right? Using the microscope. So the red color would be second harmonic, and then the green color would be third harmonics, right? So if you look at it here. So here's some of this is my favorite sample, which is orange cansite. If you look at this rock, you can see as if the flame has been freeze inside the rock, right? You can see the beautiful feature here. And we are using this technique right now to try to understand the how the rock will form, right? The condition, the geological condition at the moment that the rocks was formed millions of years ago. So this has become a new tool for us to study these rocks and minerals. And with multiphoton microscopy, you can get a 3D image of the rock, right? as I show here, you can go through, in this case, 2 millimeter inside the rocks, right? With the traditional technique, what you have to do is to cut the rock into thin slices, and then you have to use a microscope to look piece by piece, right? But with this multiphoton microscopy technique, you can look inside the rock without destroying it, right? You don't need even to do sample preparation that much here. So this is another rock that we use to look at quartz columns in the so-called tiger's eye minerals here. So this work my PhD student published recently in this paper here. So if you would like to learn more, then you can look at those. So next, we now look at biology now, right? So this is a mosquito that basically we just got from outside. And then you can put under the microscope and you can image the mosquito in 3D without any preparation at all, right? So the green color here, that is third harmonics, and then the red color, that would be second harmonics. So the red color shows the muscle, the bundle, the muscle of the mosquito, right? So you can see the wing area has a lot of muscle, right? So, and then if you do a scan in 3D, you can get a 3D image that you just seen on the left side there. That is basically the needle, right? So the thrombosis, that's the needle that the mosquito use to suck our blood, basically, right? The needle would go out from here and puncture your skin and then suck the blood off, right? So that the mosquito that we have. So we have looked at ants, we have looked at a lot of worms and insects. So those are really interesting things to study. So next, I will mention very briefly about brain imaging techniques that we have here in our group. So this is a cross section of a mouse brain, right? You can see the scan here is 2 millimeter, and we got this image by mosaicing thousands of images that we get, right? So you can see here the red is the endogenous fluorophore from the mouse itself, and then the blue is from the so-called yellow fluorescent protein that the neurons actually express themselves. So now if you zoom in, you can see the individual neurons, right? That you can see these are the neurons. And then you can see all the dendrites that connect the neurons together, right? And if you zoom in further, you can see this very complex structure. So you see the neurons here, and you see the dendrites, right? So now if you scan the sample in 3D, then you can get an image that looks like this, right? So this is about 300 micron by 300 micron by 300 micron dimension here. Okay, so you can see all the neuron in green, and then you see the red endogenous fluorophore. So right now, we actually have a technology that we can map out the brain in 3D, right? So we will be able to know the location of 8 neurons, and we know all of the connections that the neuron have with one another, right? So hopefully this would give us a better understanding of the brain. So with our collaborator, we're trying to use this technology, for example, to map the parasites inside brain to be able to identify different group of neurons that will be responsible for different type of behavior, right? So there's a lot of interesting things that we can do with brain imaging here. So I won't have time to go into detail. The next one, I want to go in more detail because I think it's very relevant to healthcare. And this is a project that we've been working on for a few years, and there's some success that I would like to share, which is on pancreatic cancer imaging. So this is not as common as other cancers, as you can see here. So the pancreatic cancer is about 3%, right? If you look at in the male and female population, but the problem is that pancreatic cancer is the 4th highest rate of cancer death in men and women. So this is quite a nasty cancer. You can see here in men that about 7%, right? In women it's about the same 7%. It's a nasty cancer, and the reason why it is so dangerous is that it's very difficult to diagnose it. Okay, so if you look at the prediction of different type of cancer over the year, then pancreatic cancer is projected to be the second deadliest cancer by 2030, right? So about 9 years or 10 years from now, you can see the number of deaths by pancreatic cancer even more than liver, colon, and rectum, and many other types. So it will be the second deadliest cancer down the road. Okay, so why is this such a fatal cancer? The problem with it is that more than half of the cases that are diagnosed at a late stage, right? So you can look at this paper here. You can see the statistic of it. And with this type of cancer, does not respond well to external treatments, meaning, you use chemotherapy, for example, right? Or you use different type of drugs. It's not effective there. So the current way to combat with this type of cancer is to do surgical, right? Surgery. So basically the way to do it is to cut a section of the tissue out, right? So you open the patient up, you go to the pancreas, and basically you cut a section of the tissue out, right? And then this biopsy would be looked at by a pathologist, right? So if the pathologist look at it, because of the time constraint, he or she look at only 1% of the surface area. So if the pathologist finds cancer, then the pathologist would tell the surgeon that, okay, you need to cut more, right? So basically the surgeon cut one more biopsy and then gives up to the pathologist, and the pathologist looks again. And then the process repeat again, right? Until you find no more cancer, or until the patient have so little of pancreas left that the patient can't even survive after the procedure. So you have to stop, right? So that is how it is done now, and that is why it's so fatal. So the problem in improving the outcome of the treatment is actually in finding the margin, right, between the healthy and cancerous cells, so that the statistic is that if you remove all the cancer cells, then 80% of the patients with clean margins, right, would be alive 5 years or more after the surgery. But if there's any cancer cell left, right, after the surgery, then basically almost nobody survived past 2 years after the study or after the surgery, right? So that's why the most important thing in this type of cancer during the surgery is to find the margin, right, the border between the cancer cells and then the healthy cells. So the current medical practice for surgical removal takes a lot of time, right? Take tissue biopsy, and then the pathologist have to slice the tissue sample using a microtome to make very thin slices, right, about 5 micron thick. And then you have to stain it with H&E, right? And then you have to look at it, right? So it takes about 30 minutes or so to do all of this. And you can basically rely on the pathologist to provide feedback to the surgeon, right, to cut more or to stop, right? And the problem is that only 1% of the total area is examined, right? So in 1%, if you miss the cancer cell in the 99% of the area, then the surgeon would basically close the patient up, and then the patient go home with its cancer cell remaining, and the survival rate is really low there. Okay. So basically, we have shown that multiphoton imaging removes the need for stains. We have taken fresh tissue from the pancreas tissue, and we image with our multiphoton microscope, and it turned out that we get really nice contrast, right? So this is H&E staining, and this is a multiphoton image here. With H&E staining and traditional microscopy, you can look at only one surface, right? With multiphoton microscopy, you can look at multiple surface. So you don't need to do slicing there. You don't need to do staining. You don't need to slice the sample into thin slices. So that's why with multiphoton microscopy, we think that we will speed up the imaging speed a lot more. And instead of looking at only 1% of the surface area, we can now look at 100% of the surface area. And we would know for sure that there would be no cancer cell left in the patient, right, after the surgery. So this is another image that show we can identify the margin of the cancer cells and then the healthy cells very clearly. So if you look here on the top, right, that's all the healthy tissue. So you can see very clear cell structure of the pancreas. But then at the bottom, the bluish color part here from this region down, it looked more like blue, a lot of third harmonics that indicates a lot of nuclei in the pancreas here. So if you look at the border here and look at the signal of different channels here through the margin, right, through the boundary, you can see very different behavior of the signal. For example, if you look at the second harmonic, right, which is the green color here, it turned out that at the boundary between the normal cells and then the cancer cell, we have a lot of collagen, right, which show a lot of second harmonic signal that we have here. So basically, through this story, we have proved that we can use multiphoton microscopy to look at cancer margin here. Okay, so that my last slide. I hope I'm within the time. I'm not sure when should I stop here, but I would like to thank all the funding agencies that have provided funding for us to do our work, and I would like to also to thank all of my students for all the hard work that they have done in the lab, and I would like to thank you for your attention. And I was told that I have to leave about 10 minutes, right, for questions. So I hope that is about the time here. So please go ahead and ask your question if you have any.
Presentation. Thank you so much, sir, for such a wonderful presentation. I now open the session for any questions. Yeah.
Professor, it was a very nice presentation on multiphoton imaging. Compared to the conventional staining methods, how much time does it take to take this whole slide imaging of this pancreas?
So right now with multiphoton microscopy, we can do as fast as video rate, meaning it's about 30 frames per second, right? So in the current technique, it would take about 30 minutes, right, to look at about 1% of the surface area of the biopsy. Our goal is to be able to look at 100% of the biopsy within that 30 minutes, right? So then we can provide feedback to the surgeon within the 30 minutes that if he would need to go in and cut more of the pancreas, right? Or if we have been able to remove the full clean margin, and the survival rate of the patient would increase significantly, right? From the data, if we have removed all the cancer cells, and 80% would live more than 5 years after the surgery, right? So, so that's the goal that we would like to achieve.
Is there also any autofluorescence, because you are seeing multiple signals? is this anything that we need to do for the signal processing as such?
So the image that I showed you earlier, that all endogenous signal, right? So second and third harmonics, that's an inherent signal from different structure of the tissue. But then we have 2-photon excitation fluorescence. And those are the endogenous molecules like NADH and things like that, proteins, right? And then we also have 2-photon excitation fluorescence. So these images were taken directly from the sample without any processing. So basically, we did not even stain or put any fluorophore into the tissue at all. So that's why we think that we would save a lot of time doing the imaging task here.
Thank you very much. It's a very impressive work.
Thank you for the good question.
I have a small question. Since we are dealing with multiphoton imaging, multiphoton processes, so generally these are highly intense process. So how do you deal with the sample degradation that can be a possible challenge when you're imaging systems, because multiphoton highly intense process can tend to damage the samples?
Yeah, thank you for the question. It's a very good question. Basically, at the beginning, when we started to do this work, that's the question we asked ourselves. We don't want to create problem when you do imaging, right? So there are two different mechanisms for damaging here. The first mechanism is based on thermal, right? When you focus the laser beam on the sample can absorb that photons, and that energy would be converted into heat, right? So if there's a lot of heat, you can burn the tissue, right? So that's the first mechanism. So in order to deal with that problem, we use femtosecond laser pulses, right? So with these laser, we working with femtosecond pulses, right? So these are really short bursts of light. So the average power that we use to do the image is in the milliwatt range. So this is low enough that you don't create a lot of heat to cook your sample, right? So that's the first mechanism for damaging. So the second mechanism is based on the high field intensity of the laser beam, right? So the laser beam has an E field. The E field can be strong enough that you can tear the electron out of the atoms, right? You can ionize basically the atoms or the molecule in the sample. And that ionization process can damage your tissue, right? It turned out that we are far away from that. So we use the electric field of the laser beam. We push the electron out of equilibrium, but we don't push it far enough that it would be ionized, right? So we are way below the damage the sample, following this mechanism. So I think, in fact, in Europe, they have already approved the use of multiphoton imaging or microscopy already. If you use output power from the laser less than about 50 milliwatt or so, which is typically the case that we are doing with our microscope. Okay. So I hope I have answered your question there.
Yes, sir.
It's actually a very good question. Yeah.
And just another curious question, maybe two parts to it. One, you told that you had used an 80 femtosecond laser centered roughly around 1550 nanometers, 1.55 microns. So as I understand, that would require a bandwidth of almost the entire C band. So how do you make such ultra short pulses to that, say something like 80 femtoseconds, sub 100 femtoseconds? And the other part of the question is, which spectrum range would be much better for such an application? For example, in the 1550 region, you would have the water absorption peaks disrupting your entire system. So is there a better region? Is 2 micron better or 1 micron better? Which region of the spectrum would be a better source for the fiber lasers?
That is a very good question indeed. So I would answer the second part first, okay? It is actually a very important question to understand what would be the best laser wavelength or spectrum region that we can work with, right? To answer that question, I think the first thing to do is to look at the optical properties of biological tissue, right? So in biological tissue, most of the time we have water, right? And then we have a lot of scattering because of the inhomogeneous structure of the tissue, right? So with water absorption, we actually have transparency window up to about 1.3 micron. And in the visible up to about 1.3 micron, and then we have a peak at about 1.4 micron, and then it goes down to about 1.7 micron. That's where the dip of the absorption of water will be, and then after 1.7 it goes up very fast, right? Because of this vibration on already of water molecule, right? Of the overtone absorption. So the answer. Which wavelength would be best? My answer would be it depends. So first of all, a lot of time people actually use fluorophore, right? It's very specific fluorescent protein that they need to excite, and those actually have very well-defined wavelength that you need to use, right? So a lot of time, because of the availability of TISAB laser, people use 800 nanometer, right? But now if you use something at short wavelength, then the scattering is really high, right? So scattering scale with 1 over lambda power 4, right? So that's the Rayleigh scattering. So we would like to go to longer wavelength in order to go deeper, right? But you don't want to go too long because you would hit absorption of the overtone, right? So right now, in order to go very deep in the tissue, people prefer to use 1.7 micron, right? In order to go deeper, or about 1.3, and in our case we use about 1.55, and indeed we have to deal with water absorption there, but it's not at the peak, right? So in our lab we have developed a laser working at 1.7. I didn't mention it here for the sake of time. We have developed fiber laser working at 1.3 for deep tissue imaging, and then for the brain imaging work, for example, we use the laser working at about 1.03 micron based on ytterbium. And that is a laser that you can excite a lot of endogenous fluorophore. And that's a laser that we use to do the pancreas imaging works that I showed earlier. Okay, so hopefully that part is clear. The first part on the bandwidth of the laser, indeed, if you have 100 femtosecond pons at about 1550 nanometer, you have about 35 nanometer bandwidth, right? It turned out with fiber laser it's not difficult to get that type of bandwidth because we have a lot of nonlinear response inside the fiber itself. So we can use the fiber as a nonlinear broadening medium, right? So you can start with something at about 10 nanometer bandwidth, 300 femtosecond, and you can go through a special fiber and you can broaden it down to out to even 40, 50 nanometer, and then you can compress it back to I think the shortest that we made with 1550 nanometer laser is about 50 femtosecond, right? It's even much less than 80 femtosecond that you mentioned earlier. Okay, so hopefully I answer your question.
Yes, sir. Thank you so much.
Any comments or any other questions that you have?
Yeah, there are a couple of questions from participants virtually. So one of the question is, what is the spot size of your focused beam? When you
That's a very good question. So the laser spot size depends on the objective that you use, right? And ultimately it depends on the NA of the objective. So the best objective that we have in the lab would have about 1.3 NA, right? So it's a so-called oil immersion lens that we use. So now if you use a 1550 nanometer laser, then the spot size would be about 800 nanometer or so. It's about lambda over 2. If you use a 1 micron laser, then the spot size in X, Y, and Z, let's say, would be about half a micron cube, right? If you think of it that way in 3D. So now that's the laser spot size. The resolution depends on the nonlinear process that we use as well, right? So if you have a second harmonic or second order nonlinearity to work with, then your resolution is the laser spot size roughly divided by square root of 2. If you use third harmonic generation process, then the resolution would be the laser spot size divided square root of 3, right? So in principle, we can get down to about maybe 300 nanometer or so resolution of the system.
Yes, sir. Thank you so much. It's just another question, probably the last question for this session. What are the ratios of probabilities of the fundamental, the second harmonic and third harmonic excitations to occur for a living healthy cell versus a cancerous one? What are the ratios of probabilities for?
That is a very good question. So I don't think I have a number from the top of my head. What we have observed is that in cancer tissue, typically we have a lot of nuclei, right? And then the nuclei typically is denser compared to other material because cancer cells, they just multiply, right? And that's why you have a huge amount of cells in a small space. So what we have observed is that the third harmonic signal is really, really strong there, but I don't know how many times stronger. There's a graph that I show in one of the slides. Maybe you can look at that. You can see the ratio there. I don't remember from the top of my head. Another interesting thing is that at the boundary between the cancer cell and then the normal cell, we saw a lot of second harmonics or collagen, right? So these are the fibrous tissue that's present. So when we talk to the surgeon, for example, what they do, without any imaging guide technique, the surgeon actually use his hand, right, or her hand to feel the tissue, right? And then the cancer cell typically is harder or something. And then based on that feel, you can do the biopsy, right? And I think we confirmed that with multiphoton microscopy because in the cancer tissue, the density of cells is really high, and we saw a lot of nuclei of cells there. So it's a very interesting question, and we should look into the quantitative numbers there.
Thank you so much, sir. Thank you so much for coming over and presenting this beautiful, inspiring talk. We look forward to listen to you more and connect with you in the future. Thank you so much, sir. Thank you.
Yeah, thank you for your invitation.
Thank you, sir.
Thank you.